All right, buckle up for our deep dive today. We're going to help you crush that upcoming exam on antibiotic resistance.
You know it.
We'll be focusing on bacteria and their sneaky ways of dodging these drugs. And we'll be drawing insights directly from Walsh and Wensvich's book, Antibiotics, Challenges, Mechanisms, Opportunities.
Exactly. We'll unpack some of the key concepts, get into some seriously interesting examples.
And of course, hit you with those exam-style questions to make sure you're ready for anything.
Sounds good to me.
So you're already familiar with antibiotic resistance, right? It's a big deal in the clinic.
Yeah.
It's basically bacteria's superpower to survive and grow, even when they're hit with antibiotics that should be stopping them in their tracks.
Right.
But there are, like, important shades of gray within that definition, right?
Absolutely. There's a key difference between, like, intrinsic resistance and acquired resistance.
Okay, break that down for me.
So intrinsic resistance is kind of like a bacterium's natural armor.
Okay.
It's just part of their structure or function.
Mm-hmm.
Take gram-negative bacteria, for example. They have that outer membrane, which is like a fortress wall. It's naturally tough for some antibiotics to penetrate.
So it's like they're born with a shield.
Yeah, exactly.
Yeah, makes sense. But what about bacteria that weren't born tough, but became resistant over time? That's the acquired resistance, right?
Exactly. Acquired resistance. This is the real game changer, especially in clinical settings. It's driven by genetic changes that turn a once susceptible bacteria into a resistant one.
And that makes it tricky to treat, right?
Oh, absolutely. It's a constantly moving target.
Like an ongoing arms race.
Exact.
Bacteria are constantly finding new ways to outsmart our antibiotics.
Yeah.
Speaking of outsmarting, Walsh and Wensvich break down 4 major mechanisms bacteria use to resist antibiotics. It's like their toolbox of survival tactics.
Yeah, it's like let's start with efflux pumps. These are like little bouncers at the door of the bacterial cell.
Okay.
They pump antibiotics right back out as quickly as they enter.
It's so frustrating. Clever, but frustrating. What other tricks do they have up their sleeves?
Well, there's antibiotic modification or destruction. Some bacteria produce enzymes that can either break the antibiotic down or change it in a way that makes it useless.
Sabotage.
Yeah, exactly.
Fine, what else?
Then there's target alteration. This is where bacteria get sneaky and actually change the target site that the antibiotic is supposed to bind to.
Oh.
It's like they're changing the locks on their doors so the antibiotic key no longer fits.
So they're basically making themselves invisible to the antibiotic. What about gram-negative bacteria with their fortress-like outer membrane? Do they have any other tricks?
Oh, they do. They can modify those outer membrane porins, which are like gatekeepers controlling what goes in and out of the cell.
Right.
By changing these porins, they can make it much harder for antibiotics to get inside.
It's all about controlling access then.
Yeah, keeping antibiotics out or changing their target so they can't do any damage.
Okay. Now, this is interesting to me. If some bacteria actually produce antibiotics themselves, how do they avoid poisoning themselves?
Right, that's a great question. It really shows how ingenious these microbes can be. Antibiotic-producing bacteria have developed their own self-protection mechanisms.
So, like an antidote to their own poison.
Pretty much.
What kind of strategies do they use?
One common one is having modified target sites within their own cells. For example, their ribosomes might be slightly different, so the antibiotic they produce doesn't affect their own protein synthesis.
Like a built-in safety mechanism.
Exactly.
What else?
They can also use efflux pumps to keep the concentration of the antibiotic low inside their cells.
So kind of like a ventilation system to get rid of any toxins.
You got it. A built-in detox system.
Pretty impressive.
Yeah.
Any specific examples?
Oh, tons. Thiocillin producers, for example, have these modified ribosomal subunits that make them resistant to their own antibiotic. And then there are the phenamycin producers. They have a beta-lactamase enzyme that neutralizes any excess carbapenem that builds up in their cells.
Wow. So they're constantly modifying their defenses and weapons.
Yeah.
But let's shift gears a bit. Let's talk about acquired resistance. How do bacteria get their hands on those resistance genes in the first place?
Well, acquired resistance usually happens in one of 2 ways. Vertical gene transfer or horizontal gene transfer.
Okay, I'm listening.
Vertical gene transfer is pretty straightforward. It's just basic inheritance. Resistant bacteria pass those resistance genes down to their offspring.
Survival of the fittest, bacterial style.
Exactly.
Okay, what about horizontal gene transfer?
This one's more like sharing notes in class. It involves bacteria sharing those resistance genes with each other, even across different species.
Bacteria with social lives. Okay,
Yeah.
but how does that actually work?
There are a few ways. First, you have transformation. Bacteria can actually pick up stray DNA from their environment, maybe from a dead bacterium nearby.
So they're like scavengers too.
Pretty much. Then there's conjugation. Bacteria can connect to each other and directly transfer genetic material, including resistance genes.
So like plugging in a flash drive.
Exactly. And finally, there's transduction. This one involves viruses, specifically bacteriophages, acting as delivery trucks for these genes.
Oh, wow. So we've got bacteria picking up genes from their environment, sharing them with their buddies, and even getting deliveries from viruses.
It's a whole network for sharing resistance.
And that makes it a tough problem to solve, right?
Exactly. Acquired resistance isn't just about one bacterium becoming resistant. It's about that resistance spreading throughout the entire population.
I'm starting to see why this is such a big deal. And we've only just begun to scratch the surface here.
There's a lot more to uncover. Some bacteria get even more creative with their resistance tactics.
Oh, how so?
They can actually change their shape or behavior to become more resistant.
Change their shape?
Yeah. Think of persister cells, for instance. These are like bacterial sleeper agents. They enter a dormant state, kind of like playing dead. This allows them to evade antibiotics that target actively growing cells. They're sneaky.
And that's not all, is it?
Nope. Then there are biofilms. These are like bacterial cities where bacteria group together and form these protective communities.
So they're not just lone wolves.
Not at all.
Interesting. What else?
This might blow your mind. There's this concept called the resistome. It's basically a giant library of resistance genes out there in the environment just waiting to be tapped into.
A library of resistance.
Exactly.
Where does it come from?
All sorts of sources. Antibiotic producers, soil bacteria, even potential precursor genes that bacteria already have for other functions.
So there's this whole reservoir of resistance genes just sitting out there waiting to be picked up. That's a bit scary, honestly.
Yeah, it makes dealing with antibiotic resistance even more complex.
Okay, we've covered a lot about how bacteria become resistant.
We have.
We've talked about mechanisms, the resistem, and even examples like linezolid and quinolone resistance. It's been a crash course in bacterial warfare.
It has.
But before we move on, should we dive into some concrete examples from Walsh and Wensvich, like linezolid and quinolone resistance?
Just do it. Solidify our understanding a bit.
Sounds good to me.
So linezolid is interesting. Bacteria mainly resist it through mutations in their 23S rRNA, which is the target for this antibiotic.
So it's back to that changing the locks tactic.
Exactly. These mutations mess up the binding site, making the antibiotic useless.
Makes sense. What about quinolones? How do bacteria resist those?
Quinolones are a bit more complex. Bacteria have a whole arsenal of ways to fight them.
Okay.
Target mutations, efflux pumps, plasmid-borne resistance genes, even porin loss in those gram-negative bacteria.
So they really pull out all the stops. It's amazing how resourceful they are.
It really is. They're incredibly adaptable and resilient.
Well, we've covered a ton of information about antibiotic resistance.
For sure.
From basic mechanisms to the resistem to those specific examples like linezolid and quinolone resistance. Are you ready to test your knowledge with some exam-style questions?
Absolutely. Let's see what you've got.
Bring it on. I'm feeling pretty confident after this deep dive.
All right, here's your first question. What are the 4 major mechanisms of bacterial antibiotic resistance?
Okay, I think I've got this. Let me see. We've got efflux pumps, antibiotic modification or destruction, target alteration, and outer membrane porin alteration. How did I do?
Nailed it. You've been paying attention. Okay, question number 2. What's the difference between intrinsic and acquired resistance? And give me an example of each.
Okay, so intrinsic resistance is like having built-in defenses. It's part of the bacteria's natural makeup.
Right.
Like that outer membrane in gram-negative bacteria.
Good example.
Acquired resistance is something bacteria pick up over time.
Right.
Through things like mutations or gene transfer. A good example of that would be methicillin-resistant Staphylococcus aureus or MRSA.
Perfect. You got it. Let's move on to question number 3. How do antibiotic-producing bacteria protect themselves from their own antibiotics?
This is where those clever self-preservation strategies come in.
Go on.
They can use efflux pumps to keep the internal antibiotic levels low.
Right.
Or modify their own targets so the antibiotic can't bind. Or even produce enzymes that deactivate their own antibiotic, like thiocillin producers. They have those modified ribosomes that make them resistant to their own antibiotic.
Fantastic explanation. You're on a roll. Ready for question 4?
Hit me.
Okay. What are the 3 main mechanisms of horizontal gene transfer?
Okay, let's see. We've got transformation, where bacteria grab DNA from their surroundings.
Right.
Then there's conjugation, that direct transfer of genetic material. And finally, transduction, with those phages acting as delivery vehicles for genes.
You got it. You're really remembering those key details. And my last question for this section. What is the resistem, and why should we be concerned about it when it comes to antibiotic resistance?
The resistem is like that massive library of resistance genes that exists out in the environment.
Right.
It's a concern because it's a huge pool of potential resistance genes that bacteria can access and share. That makes it even harder to stay ahead of antibiotic resistance.
You aced it. You've got a great understanding of the resistem and why it matters. We'll pick back up in part 2 of this deep dive with more questions and a deeper look at the challenges and opportunities in the fight against antibiotic resistance.
Back again for more on antibiotic resistance. Last time we really got into those different ways bacteria can become resistant.
Yeah.
But Walsh and Wensvich also bring up some really interesting points about the resistem. This massive collection of resistance genes just hanging out in the environment. Kind of scary, right?
It is a pretty sobering thought. That resistem represents this huge potential for resistance to top up.
Even to antibiotics we haven't even developed yet.
Exactly. It's like bacteria have this secret weapon stash just waiting to be used.
But how does nature itself handle antibiotic resistance?
Yeah.
I mean, antibiotics are naturally produced by a lot of microbes out there.
Right.
Shouldn't resistance already be everywhere?
You'd think so.
Yeah.
It's a good question. And it really gets to how we humans are impacting things. Walsh and Wensvich point out that in natural settings like soil, those antibiotic levels are generally pretty low and they change over time.
Okay.
That kind of dynamic environment might actually help prevent that, that runaway selection for resistance that we see in hospitals.
So it's almost like our overuse of antibiotics in healthcare is messing things up.
Yeah.
Creating this intense pressure that's just driving resistance to evolve and spread like wildfire.
Right. That constant exposure to high levels of antibiotics, especially in hospitals, gives those resistant strains a big advantage.
Makes sense. They can outcompete the susceptible bacteria, so the resistant ones take over.
Exactly. So what do we do?
Well, what about synthetic antibiotics?
Yeah.
Could that be a way to outsmart these resistant bacteria? I mean, if they haven't seen these molecules before, maybe resistance will be slower to develop.
That's the idea behind synthetics, yeah. Introduce something completely new.
Okay.
Throw a curveball they're not ready for. We might gain a little bit of an edge, at least for a while.
But bacteria are pretty good at adapting, right? It's probably just a matter of time before they figure out how to resist those new drugs, too.
You're not wrong.
Okay. So synthetic antibiotics might buy us some time, but they're not a permanent solution. Speaking of time, Walsh and Wensvich talk about linezolid.
Yeah.
It's had surprisingly low resistance levels compared to other antibiotics. What do you think that is?
Linezolid is interesting. It has this unique oxazolidinone structure that bacteria haven't really encountered much in nature.
So it's new to them.
Yeah. And that seems to be making it tougher for them to develop those resistance mechanisms.
So novelty is key, but bacteria are clever. They'll probably figure it out eventually, right? Ugh, this is all starting to feel a bit overwhelming, like we're fighting a losing battle.
It's definitely a challenge, but I wouldn't say it's hopeless. There are things we can do to slow down the spread of resistance.
Okay, I need some good news right now. What can we actually do to turn things around?
Well, one of the most important things is antibiotic stewardship.
Okay.
We have to be much more careful about how we use antibiotics.
Right.
Make sure they're only prescribed when they're truly needed.
No more antibiotics for every sniffle.
Exactly. Preventing infections in the first place.
Okay.
Good hygiene practices, promoting vaccination, strong infection control in healthcare settings, all of that helps.
So it's about reducing the need for antibiotics altogether.
Yeah.
It sounds like we need a multi-pronged approach.
We do. And of course, research into new antibiotics and other kinds of therapies is crucial. Scientists are looking at all sorts of new approaches, from targeting those bacterial virulence factors to developing phage therapy, which uses viruses to kill bacteria. It's about finding new weapons in this fight.
So we need new weapons, better defenses, and a smarter overall strategy. But are we all on the same page about this?
That's a really important point. We need to raise awareness about this issue.
Yeah.
Educate people about the risks of overuse, the importance of finishing those prescribed courses, and the simple things they can do to prevent infections.
It sounds like we need a global effort to tackle this.
We absolutely do. Antibiotic resistance is a global threat, and it needs a global response. Collaboration, innovation, and commitment are what we need.
Well, this has been incredibly insightful, but I'm ready to put my knowledge to the test. Ready for those exam questions.
Bring them on.
We're back for the final stretch of our deep dive on antibiotic resistance. Bring on those exam questions. I'm feeling ready.
Awesome. All right, let's jump into some scenarios you might see on the exam. Picture this. You're in the lab, you've isolated a bacterial strain, and it's resistant to multiple antibiotics.
Okay, yeah.
How would you figure out if that resistance is intrinsic or acquired?
Let me think. Well, first, I need to know what species of bacteria I'm dealing with.
Right.
If that species is already known to be resistant to those specific antibiotics, that's a good sign it's intrinsic, all right?
Makes sense.
I could check resources like Walsh and Vensvich, see what they say about that bacteria.
Smart move. Knowing the species and its usual resistance patterns can give you a head start.
Yeah, exactly. Then I'd want to look at the genome of this resistant strain. Compare it to a susceptible strain of the same species.
Okay.
If there are some genetic differences, like mutations or extra genes in the resistant strain, then that points to acquired resistance.
Good thinking. Genetic analysis can really tell you a lot about those resistance mechanisms.
And to be extra sure, I could try exposing the bacteria to different concentrations of the antibiotics, see how it grows. If the resistance patterns match up with what we know about things like efflux pumps or target alterations, that would be more evidence.
That's a really thorough approach. You're combining genetic analysis with what you observe, which is a great way to understand that resistance profile.
All right, I'm ready for the next challenge.
Okay, let's switch to a clinical scenario. Imagine you're treating a patient with a bacterial infection.
Okay.
The first antibiotic treatment doesn't work. What are some reasons why that might happen, and how would you figure out the root cause?
Okay, this is where things get interesting. Well, the first thing I'd consider is that the bacteria might have developed resistance during the treatment.
That's a big one. Happens more often than we'd like.
It could be through mutations or acquiring resistance genes.
Right. Acquired resistance during treatment is a real challenge.
I'd also want to make sure the patient was taking the antibiotics correctly. Right dose, right length of time. Non-compliance can mess things up.
Absolutely. Patient adherence is crucial for successful antibiotic therapy.
Another possibility is that the initial diagnosis was wrong.
Oh, yeah.
Maybe the patient has a different infection, one that isn't susceptible to that antibiotic.
Very important point. Getting that initial identification right is key.
So to get to the bottom of it, I'd start by getting a new sample from the patient.
Okay.
And run some susceptibility testing. That'll tell us if those bacteria are resistant to the first antibiotic.
Makes sense. Susceptibility testing helps guide treatment decisions.
And if they're resistant, I'd want to know how. What's the mechanism? We could do genetic analysis or some more testing. Knowing that will help us pick a better antibiotic.
That's strategic thinking. Tailoring treatment based on the resistance mechanism is key.
And of course, I'd always check the patient's medical history, make sure they're taking their medications correctly, just to rule out anything else.
Always good to have that full picture.
Okay, what's next?
All right, let's zoom out a bit and talk about the bigger picture. How does the resistem make it harder to develop new antibiotics?
The resistem, that library of resistance genes, it's a real headache for drug development.
Yeah.
Because even if we come up with a totally new antibiotic, there's a chance that a resistance gene for it already exists out there.
Exactly. That's the worry. The resistem is so vast.
So our new antibiotic might work great at first, but resistance could pop up sooner than we want if those genes start spreading.
It's an arms race against those microbes. The resistem just adds another layer of complexity.
So we need to be thinking ahead.
Yeah.
Trying to anticipate those resistance mechanisms when we're developing new drugs.
We can't just focus on short-term fixes. We need long-term solutions.
Hit me with another question.
Okay, this one's all about public health. How does horizontal gene transfer contribute to the spread of antibiotic resistance, and why is that a problem for us?
Horizontal gene transfer is like a superhighway for those resistance genes.
Yeah.
It lets them spread really fast, even between different species.
It's like a social network for bacteria sharing their survival tips.
And that's a problem because a resistance gene that pops up in one bacteria can jump to others.
Right.
We could end up with these multi-drug resistant pathogens that are super hard to treat.
And those transfers can happen between harmless bacteria and pathogenic ones. That creates new threats and makes existing infections harder to manage.
Like a domino effect. Resistance emerging in one spot can have huge consequences.
Exactly. We need to understand how this transfer works if we want to control the spread of resistance.
Okay, I'm ready for the final challenge. Give me your best shot.
All right, last question. Antibiotic resistance is a growing threat. What are some things we can do to make sure the antibiotics we have keep working and to develop new ones.
This is the big question, isn't it?
It is.
We need to do a lot of things. First, we have to use antibiotics responsibly.
Absolutely.
Cut down on unnecessary prescriptions, make sure patients finish their courses, use the right antibiotic for the job.
Antibiotic stewardship is crucial. These are precious drugs.
And we need to invest in research. New antibiotics, new therapies. We can't rely on the old ones forever.
We have to keep innovating.
Preventing infections is also super important.
Yeah.
Better hygiene, more vaccinations, stricter infection control in hospitals.
Prevention is key.
We also need to educate people.
Right.
Make sure they understand how serious this is and what they can do to help. Good hygiene, vaccinations, responsible antibiotic use.
An informed public is a powerful tool in this fight.
And we need to work together globally. This is a worldwide problem.
It is.
We need everyone on board, governments, healthcare organizations, researchers, sharing information, resources, and strategies.
It's a challenge that goes beyond borders. We need a unified response.
Wow, this whole deep dive has been eye-opening. Antibiotic resistance is a huge issue. There's still so much work to do.
There is.
But I feel hopeful. I think if we work together, we can find ways to protect these life-saving drugs for the future.
I agree. Knowledge is power, and I'm glad we could help you understand antibiotic resistance better. Keep learning, keep asking questions, and keep spreading the word about responsible antibiotic use.
This has been an amazing journey into the world of antibiotic resistance. Thanks for guiding me through it. I feel prepared to face this challenge head-on.
It's been my pleasure. Remember, this fight doesn't end here. We all have a role to play.
That's a great reminder. Thanks for joining us for this deep dive into bacterial antibiotic resistance. Until next time, stay curious, stay informed, and stay healthy.